make that work very easy. Just you can submit the sequence. They have already deposited nearly, I think, more than 20,000 proteins in the databases. This AlphaFold is a collaborative work of this EMBL and Google. So they are using deep deep mind, I think, the algorithm. So it is very accurate. Nowadays, scientists are using this database. So you can go to the directly visit the AlphaFold database. You can type the query sequence or name. You can just search it and download the structure of the protein, query protein, right? Before that, downloading, the page itself has so many informations like a confidence level. Confidence level means nothing but how much that structure is reliable, how much we can believe that structure is good or not, right? So if the PIDD score L, actually DD score is more, means that is more reliable, and more than 90 means high confidence. High confidence means you can that is structure is very good, and 70 to 90 is good model. You can believe that, you can use for further research. And because we are producing a structure, we are modeling a structure by using the sequence, amino acid sequence. So we have to analyze that whether the structure is good or how much it can be reliable, like that. So 50 to 70 low confidence, so use this with caution. You have to reanalyze the structure. If less than 50 means 50 and less than 50 means you have to reanalyze the structure, and you have to correct it, and you can work it. So predicted alignment error also you can see. So in this you can see the domain details, how many domains are there, and where the domains starts, what are the starting amino acid and which is the ending amino acid, like that, and what is the position also you can see, and the quality score and length also. You can see everything is shown in the page. You just go and copy that or take a snapshot and produce it as a result. And here you can also find pathogenicity details of the protein you have modeled, right? See that likely to be pathogen of missense amino acids, and the what is that in the structure? How many amino acids are possibly get missense mutations and involved in the pathogenity diseases, right? So at last, you can download the respective PDB structure. So you for that you can compare with the model structure and PDB structure. PDB means the structure which is deposited in the PDB, which is resolved by, which may be resolved by, or no, it should be resolved by X-ray or NMR. X-ray is better, you know that. And shall we go to the experiment now? Alphafold, just you can click here, you can go that. Instead of that, I can type. So I can type Alphafold. You can just directly go to the second option, to the structural database, and see that you can paste your gene of interest or protein sequence and the name, whatever it may be. Then instead of uh, searching a sequence, I'm going to search the sequence from the what uniprot. So uniprot, I just want to know about the hemoglobin. For you, you have already a protein, right, for the bioinformatics laboratory. So you can just use that sequence of the protein. So just I am going to select a hemoglobin subunit. Again, I need only the human 208 entries, and I need a reviews. So 66 entries. So this is the 147 amino acid is here, 122 also is there. So I need the first one. For the practical purpose, I'm just seeking this thing. So if you want, you can use your own protein of sequence. I hope you already had that, so you can directly put into the search box. right? And I need a sequence here. Sequence is there, and just I have download this thing. I can copy. And most important thing is here we need not to use any FASTA symbol here. And this is a FASTA symbol, right? That greater than symbol is called as FASTA symbol. That FASTA symbol is not need not to be put in this query box. So you can copy from the first amino acids. So I can easily find that. So I copy that and paste here, and I have given search. Right, is it running? Now, after getting the results, you have to filter. Means like Homo sapiens again. See that Homo sapiens, there are 36 entries are there. Previously, it took me nearly 20,000, I think, 2,000 or 20,000. And now it became only 20. Among the 20, I need only that reviewed protein by the Sysprot. See that only 5 entries are here. Among them, I am going to select based on the PLDTT score, right? So if you want to know what is meant by PLDTT, you can see that per residue measure of local confidence, right? Which means the confidence level means the possibilities of more possibilities of the each and every amino acid in that position. So if the score is high, the protein structure also high. We can believe that. So this is 97.06 and here is 96 only, and here also what is that? The score is 97 again, 97.028. So it is highest, and then here 97.18. So you can choose any 97. um, for example, I am going to choose 97.28, which is the epsilon n, right? This is the highest one among them. So I just click here. See, you can find the structure of the protein. You just take a snapshot of this window. You can put it as a result. So you can see sequences. If you place cursor where is the position of the sequences has shown. And the first thing, the model is highly confidential, which means more confidence is there because of this blue color, and light blue indicates less than 90, more than 92. What is that? Score is less than 90, right? Okay. And here you see that there is the only one amino acid which is in the color, the light blue color, the second amino acid, I think, second amino acid which is valine 2, and the yellow color also is here, that is very low, sorry, low, is low things. There is only two amino acids are present as an high and low. Otherwise, all the amino acids are at the position, exact position. So the model confidence is high, very high. So we can believe the structure and we can get it for the further analysis. Okay. If you want, you can find which amino acid it is and which chain it is. Okay. By placing the cursor on the model. You can rotate, you can search, right? You can do all the graphics work here. The second one is you can find the missense pathogenicity mutation and see that likely begin this color, and this is uncertain, and this is likely to be pathogenic. The color maroon color are likely to be pathogenic. Say that these amino acids are likely to be pathogenic. You can find the information of amino acids. If you have only few amino acids, means you can list out the amino acids which are possible to missense mutation with pathogenicity. Otherwise, if you find more than 10, means you leave it, just show the picture alone. Okay, as a result. And here next thing is domain information. You can just click that, you can find the domain 1 is there, and the only one domain is present in that, right? So if you click here, you can find the domain name. So I will click, see that I will click here and find the domain name, domain 1, and score is 96, that is high confidence level. Then boundaries are fifth amino acid to ninth amino acid. This is fifth amino acid to ninth amino acid, 19th amino acids, and again it starts with 63rd amino acid to 142 amino acids. The total length of is 95 amino acids, and average PLDT score is 96.63, 97.63. So you can believe this structure for the further process. Okay, this is alpha missense pathogenicity heat map. At present, we don't use that. You need not to put any screenshot on this, right? So you can go for next one. The next one is about similar structures. This is what we need. Similar structures, this is actually what we got in the window is predicted structure, right? Predicted by using the sequence information, right? And here you can find the similar structure of the PDB, which was already deposited in the PDB by conforming by the X-ray or NMR. So you can choose based on the e-value. E-value should be less, very less than 0 or more negative is the best structure you can get. Sequence identity is 100. This is the resolution in Armstrong. This should be less than 2, but it is not available here. This 1.90 is here, but the query is sequence similarity is 75.3 only. So see that sequence identity is only 75 point. So we cannot, and the e value also big then comparatively. This is minus 17 and this is minus 16. So we can believe that we can choose any one of the structure of PDB. So almost the first structure will be the good one. So you can just mention, take a snapshot and mention this. What is the structure of those things? Just click it and you can see subunit epsilon, the structure and everything PDB. Now this is PDB in Europe, PDBE. You can also check in the PDB, right? This is what you have seen in the PDB itself. See that some ligand is there, hemoglobin beta chain, maybe ferrosulfuric, protoferrin 9 containing Fe is there. So you can just, this is the corresponding sequence of what is that, a hemoglobin, which we have modeled in Alphafold. You see that there are 4 chains in that, I think, but we have, this is the sequence what we have given, right? So it can be differentiated by using colors. So this is about your Alphafold experiment. Then at last you have to download the PDB structure here. So the PDB file, you can download this file just clicking and the download will be happened. So you can just view that. It's Alphafold monomer, right? Prediction. See that prediction. This is a predicted structure, but it is more reliable because the accuracy is high. So predicted structure. At the same time, you can download the PDB from the PDB that is retrieved from submitted through the lab experiment. Okay, laboratory experiment. This is about the experiments. You can submit the PDB file also as a result. Okay, thank you.